Folate's relationship with the gut microbiome is a remarkably nuanced and underexplored aspect of human health, revealing how this vitamin not only nourishes the host but also participates in a two-way dialogue with the trillions of microbes inhabiting the digestive tract. While folate is typically associated with systemic roles such as DNA synthesis, methylation, and fetal development, it also plays a quiet, foundational part in the architecture and resilience of the gut microbiome. In fact, some strains of beneficial bacteria, particularly within the genera Lactobacillus, Bifidobacterium, and Enterococcus, are capable of synthesizing folate themselves, contributing to the host's folate pool. Conversely, the host's folate status can influence microbial growth patterns, composition, and even interspecies competition within the gut. This symbiotic relationship has profound implications for digestion, immune modulation, and long-term metabolic health. One of the most intriguing aspects of this connection is that certain gut microbes depend on folate or folate-like compounds for their own DNA replication and cell division, just as human cells do. In microbial ecosystems, folate availability can shape which species thrive, which decline, and how they interact with one another. Adequate folate supports the growth of folate-producing microbes while limiting the overgrowth of potentially pathogenic species that may not contribute to, or may even compete for, this critical nutrient. The result is a more diverse and stable microbiome with enhanced resilience against disruptions such as antibiotics, poor diet, or inflammation. In this way, folate acts not just as a nutrient, but as an ecological stabilizer that preserves microbial diversity and symbiosis. What's particularly compelling is that folate produced in the colon by resident microbes may contribute to local tissue health even before being absorbed systemically. Folate plays a role in maintaining the health of the colonic epithelium, the thin layer of rapidly dividing cells that forms the barrier between the gut contents and the bloodstream. These epithelial cells require a continuous supply of nucleotides to regenerate, and folate, whether dietary or microbial, is essential for this regeneration. When folate is deficient, the integrity of the gut barrier can weaken, increasing intestinal permeability, often referred to as leaky gut, and allowing inflammatory molecules and microbial fragments to enter systemic circulation. This, in turn, can fuel low-grade chronic inflammation, a known driver of metabolic diseases and autoimmune disorders. Moreover, folate influences immune activity within the gut-associated lymphoid tissue, GALT, where a substantial portion of the body's immune cells reside. Here, folate supports the proliferation and differentiation of T cells and the proper function of regulatory T cells, TREGs, which are essential for maintaining immune tolerance to the microbiota. Without adequate folate, immune responses in the gut may become dysregulated, contributing to inflammatory bowel disease, IBD, food sensitivities, or susceptibility to infections. Interestingly, a balanced microbiome not only depends on folate to thrive but also helps maintain folate homeostasis for the host, forming a reciprocal relationship that governs both gut health and immune equilibrium. Folate's influence on microbial dynamics also intersects with metabolic health. Emerging evidence suggests that folate-producing microbes may enhance short-chain fatty acid, SCFA, production, particularly butyrate, a key energy source for colonocytes and a potent anti-inflammatory compound. This suggests that folate status may indirectly support SCFA production by nurturing the microbial populations responsible for their synthesis. In turn, these SCFAs help regulate blood sugar, satiety, and fat metabolism, highlighting yet another avenue through which folate may extend its influence into obesity, insulin resistance, and systemic inflammation. There is also evidence that folate availability affects microbial gene expression. In certain bacterial species, folate levels influence the transcription of genes involved in adhesion, colonization, and antibiotic resistance. This raises the possibility that folate may act as a regulatory molecule within the microbial ecosystem, not just a nutrient but a signal, affecting the behavior and functionality of the microbiota in ways that are still being uncovered. The implications of this relationship are especially important in clinical and dietary contexts. Antibiotic use, for instance, can disrupt folate-producing bacterial populations, potentially reducing folate synthesis at a time when tissue repair and immune defense are most needed. Conversely, diets rich in natural folate, from leafy greens, legumes, and fermented foods, not only nourish the host but may promote a more robust and folate-competent microbial community. This opens the door for nutritional strategies that use folate-rich foods or probiotics with folate synthesizing capabilities to restore gut health, especially after microbiome-disrupting events like antibiotic therapy, stress, or infection. In a health landscape increasingly focused on gut microbiota, folate emerges as a quiet but powerful influencer, a shared currency between human and microbe that shapes not only digestion but immunity, inflammation, and even mood. By supporting microbial diversity, epithelial integrity, and immune harmony, folate proves itself to be far more than a basic nutrient. It becomes a bridge between species, between systems, and between the visible and invisible aspects of wellness.